Hi, we will wait another 3 minutes and then we'll start our discussions. In the meantime, if you want to write anything in the chat, you can write in the chat, or you can try to check the chapter 6 from Lela book. So there is a channel called Charles Exam. So if you just click on that channel, there is a link. If you go to that link, like click on that link, it will take to an Excel file. So if you can just download the Excel file, and then you can be able to like take the exams. It's kind of an exams, like you can read that and you can put the correct answers according to your whatever you think, and then it will tell you like is it correct or not, and if it is incorrect, like it will give you some points. Basically it's a just all the Quizlet that one of the members collected, and he tried to put in an Excel file so that everyone can take the exams and try to check how good or bad they are in the preparation level. So it's not like you have to buy or anything. It's just you have to download it. It's kind of free. He is very nice, so he just but if you if you memorizes all the Quizlet, then maybe this exam will not gonna be that much helpful. But if you read the Lela books, like if you don't understand any concept in the Quizlet, in the Charles exam, then you can check the Lela book to understand that, rather than just memorizing the particular questions for that answer. Hi, so good evening, everyone. I think Dr. Richa, if I think you made very nice notes, so I think if you can, if you wanna share your screen and discuss first, then we can discuss. Like we can try to share our thoughts on that. Um, but it will be better if you, because your note is very nice, um, and you describe everything very nicely. So um, if you want, you can speak. Otherwise, I will gonna just read your notes and then we will try to discuss on that particular topics. I think this uh, normally this LCR, SDA, and qubit replicase is not that much important. Um, and under signal amplification, branch DNA amplification, hybrid capture assays, cleavage-based amplification, these are very important. These two are very important. So if we can discuss these four things, I think will be good from this chapter. Rest of the chapters are like some of the topics are um, um, well, like Fred is one concept. Quasar, molecular beacon, these are the some important topics. Yes.
Thank you, Abu. So I can you can help me because I am not super clear with all those things, but
Yeah.
I can definitely try.
Okay. So if you want to share your screen, you can share. So
Okay,
yeah.
let me get to the handout.
Yes. So I think these topics are also I try to check some Quizlets. Um, the amount of informations we got, I think will be good.
Yes, I think so too. Where is share screen? Right here. Okay. So can you see my screen?
Yes, we can see your screen.
Okay, so what I did was I took the notes which Abu had shared before in our chat for chapter 6, and I just um, went further on it. And there is a YouTube link which I thought was very good, at least to explain what every of all of these phenomena do. So let's discuss about TMA, which is transcription mediated amplification. So in transcription mediated amplification, what I understood is that this is a single tube reaction. And in this, you have um, uh, RNA, and then you have RNA primer. Using reverse transcriptase, you're making the cDNA. And then you guys are able to see my screen, or do you want me to make it bigger or something?
We can see your screen.
Okay, so with the reverse transcriptase, it makes the cDNA. And now, using the RNA's activity of reverse transcriptase, it digests the RNA part of it. So now you have the single-stranded DNA, and now reverse transcriptase will make it into a double-stranded DNA. Now you have a double-stranded DNA template, and this is how you will, and the RNA polymerase will use this template to make more RNA amplicons. So this is the basis of it. And organisms, I think this is very important, which organisms are used for which testing. And in this, they mentioned that Mycobacterium tuberculosis in smear, if that is positive for respiratory specimen, that is testis, chlamydia trachomatis, HIV, and cytomegalovirus. Does anybody have questions about this?
Yes, so for basically for TMA, there has three enzymes we are talking about. One is reverse transcriptase, one is RNase, and another is RNA polymerase, right?
Yes, but
Okay.
the RNase H activity is inherent. So the earlier ones which used to be there, you had to add RNase H. But now I think the reverse transcriptase has the RNase H activity. So you are just putting two enzymes, which is reverse transcriptase and RNA polymerase, because the RNase H activity is in this reverse transcriptase.
Okay, got that.
And then there are two other methods, which are the NASBA, which is the nucleic acid sequence-based amplification, and self-sustaining sequence replication. Um, do you want to talk about NASBA? Is it similar?
Yes, for NASBA, like normally this is one I think common questions for MB, those who are preparing for MB, I think, or those who are preparing like they ask questions. What's the basic differences between TMA and NASBA, something like that, or what is the basic characteristics of NASBA? So the normal answer was kind of for NASBA, it was three enzymes normally it used.
And what are those three enzymes?
One is reverse transcriptase, one is RNase H, and one is RNA polymerase.
Okay, so the difference between this one, TMA, and NASBA is that RNase H is inherent activity in reverse transcriptase.
Yes, that's correct.
Okay.
Hmm.
And then there were other things in the chapter, which was whole genome amplification, emulsion PCR, and surface amplification, bridge PCR. But I think the only thing important for emulsion PCR is I think it is similar to the droplet PCR or the digital PCR, where each template is going into a single drop and amplifying there. So I did not put a lot of attention to this. As far as I understood, that there is oil and there is emulsion which is created between water and oil.
Yes, that's correct. I think um, we covered, remember, from the genomic book,
Yes.
in the sequencing, they also discuss this part, like how emulsion PCR works, because that is also related with the sequencing. So I think we will be good. In the chat, if you guys have any questions about these topics, please write, or you can unmute yourself and ask. We can discuss more on that if you want.
Okay. So next is, so there are three things. One is probe amplification. Then there was signal amplification, and there was one more, right? Transcription mediated. So we discussed the transcription mediated amplification, and now what we are talking about is you can still see my screen, right? It's shaking. I do not know.
Yes, yes, we can see your screen. If you want, you can stop sharing and then you can share again. I think,
Okay.
yeah,
Okay. Let me make this bigger a little bit,
yeah, sure.
and I'll share it again. Sorry.
Yeah, it's okay, no problem. In the chat, can you please let us know, are you preparing for MB or SMB, so that we have much more understanding? Please write in the chat if you can. Thanks.
So next one is probe amplification. And in this, I think the difference or the questions which I saw at the end of the chapter are that it is not that the target is getting amplified. What is getting amplified here is the probe which is being used. So if we just read it together, and this is from that YouTube video, which was very nice. So in this, strand displacement amplification, or SDA, is an isothermal in vitro method for amplifying a target DNA or a RNA sequence. So isothermal means it is not a PCR amplification. Isothermal means it is at a standard temperature, or it is at one temperature. For example, in PCR, you have denaturation at 95, then annealing and extension. So that all that is not happening. It is just at one temperature. Is that correct, Abu?
Yes, that's correct.
Okay.
For isothermal, I think most of the books they suggested, like scientists or companies likes isothermal, because temperature control sometimes gets a little bit problematic. That's why these kind of technologies are very much appreciated.
And I think it is also cheaper as compared to this cycling of all the temperatures. So, for example, the Abbott ID machine, which is there in our lab, which is for respiratory virus, COVID, strep, um, RSV, those are isothermal. So it is not a PCR amplification process, but it is isothermal process. So in this, there are two steps. One is the target generation, and then the exponential amplification of the target. So we will go further down. So in this one set of, so let's look at this along with discussing the text. So this is your DNA template. It is getting denatured. So the primer, there are two kind of primers over here, which is called a bump primer and an amplification primer with the nick site. So if you see over here, on the purple strand, you have two primers which are joined. So first one is the bump primer, which is going to bump the second primer. And the second one, if you see this triangle, this has an endonucleoside. So when you are amplifying, when you are extending, so this primer is getting, the bump primer is extending, and it is displacing your um, uh, primer which had the endonucleoside, and you get the amplification product one, which is right over here. And then because your the one with the endonucleoside got bumped, got displaced, Now you have a different primer, which is different. So if you see this one is red color, this is what confused me, because that, are we putting another primer? So yes, you're putting another primer over here, which is complementary to the one which is bumped. And over here, because now you have extended the strand, endonuclease will come and it will recognize the restriction enzyme site and it will cleave it. And now, because now this nick is present, what it is doing is it is again displacing the site, the strand. And once it is displaced, if there is a double-stranded DNA, and you are using the first, the displaced strand um, is used again as a target. Does that make sense, guys?
Um, yes. So what we are discussing is like there has two primer. Uh, with one primer there has endonuclease site. So when the palm one is extending, the uh, endonuclease site one will be displaced, and again uh, that endonuclease one will also gonna amplify, and on that endonuclease site endonuclease will cut and then it will displace again. This way it will amplify again and again. That's the main basic concept.
Yes. And Abu, tell me if I'm wrong. Are there two primers or three primers? So if you see in the second part of the figure, there is a bump primer and the amplification primer.
Hmm.
But if you see at the bottom, which says extension number 2 on the display strand, that is another primer. So there should be three primers, right?
Yes. According to these pictures, it should be three. But in the book, let me double check. I'm gonna um, in the chat also, or if you guys wanna uh, um, unmute yourself and tell do they have two primer or three primer, you can tell also. um, but I think for eastern displacement there has only two primer.
Okay,
But I'm gonna double check to confirm that.
sounds good. So next is in the probe amplification is ligase chain reaction. So in probe amplification, they have discussed three assays. One is the ligation chain reaction, strand displacement, and the Q beta replicase. I don't think Q beta replicase is so important, so we will just discuss this ligase chain reaction.
Yeah.
So in ligase chain reaction, what is most important is that this would only happen, this is very good test. Okay, before we go to the ligase chain reaction for strand displacement amplification, that test is used for Neisseria gonorrhea, Chlamydia trachomatis, and Mycobacterium tuberculosis. So again, genetic diseases and respiratory virus, bacteria, sorry. So for ligase chain reaction, it is used for single cell mutations, such as sickle cell anemia. So in this case, ligase enzyme is used, which is ligating the two probes. So if we look at the picture over here, there are two sets of probes. So these are four primers are present, 1, 2, 3, and 4. And these are connected, these are hybridizing to the double-stranded DNA after denaturation. So these two primers will say go to the forward strand, and these ones are going to the reverse strand. And what is happening is that once these probe anneal at 55 degrees centigrade, the ligase enzyme, which is shown in purple over here, will connect the two. And then once it is connected, this can be amplified. And then this can go further. So for example, if you have a sickle cell mutation, as you know, in sickle cell anemia, there is a single base pair change. So the probe would be very specific to that mutation. So if that mutation is not present, the probe would not be able to bind. So even a single base pair mutation can be detected. So if the mutation is there, your probes will bind, ligase will connect it, and you will get an amplified product. Any questions about this, guys?
Um, so for ligase chain, what we are understanding is um, two probes will attach with ligase,
Mm-hmm.
and then they will amplify. And how we are getting the signal?
How we are getting the signal? That's a good question. The so I don't know why I can't move my screen. Okay, I can move it now. Okay, so this probe, which you see the red part of the probe
Mm-hmm,
on the top strand,
mm-hmm,
that is connected to a immobilized to a support um, system. For example, alkaline phosphatase. So when it is connected and the ligase has the two probes have connected, and there is a reporter at the orange probe, and that's only when it is all connected. That's when it will fluoresce, and you will have an amplification. So that is why. If you see it in the book, there is a square part
mm-hmm,
of the probe, and it is immobilized, and ligase will connect the other probe only if the sequence matches completely.
mm-hmm,
So this is connected, the red probe over here is connected to the solid support.
mm-hmm,
And when ligase has connected, only when the mutation is there, that's when you get the fluorescence.
mm-hmm. Okay, then we will get the signal. Like it will
Yes,
be give
otherwise
us
it will not. So
fluorescence.
if there is no mutation,
Hmm.
um, it would be still connected, but the other probe is not connected, and you will
Mm-hmm.
not get a signal.
Got that. Got that. Okay.
And I think in all of these, they are using alkaline phosphatase and strep evidine as a reporter system.
Okay. Alkaline phosphatase and
Yeah.
okay.
So this is just from the book. I just try to put all the resources together. So if you see
Mm-hmm.
in this, they are talking about that ligase chain reaction. So if you see that, for example, over here, it showed the purple, the red strand, right?
Mm-hmm.
And over here, if you see, there is this square bracket, which is
Yeah.
where it is immobilized to the solid support.
Hmm.
Only when there is a mutation, the ligase is connecting, which is on the left part of the screen.
Hmm.
But when there is no mutation, right, over here it is T and there's A, it is
Mm-hmm.
not connecting and it is not creating a signal.
Hmm. Got that.
All right, now we will discuss signal amplification.
Hmm.
So signal amplification is not sure what is the difference between signal and target over here, but signal amplification, they have given three examples, which is branch DNA, hybrid capture, cleavage-based, and cycling probe.
Hmm.
And this is basically used for example, HIV has many genotypes,
Hmm.
and you need to know which genotype is present, which mutation is present for the treatment.
Mm-hmm.
Same thing with hepatitis C genotype. There are multiple genotypes. And for example, I would say this is what they tested for different COVID tests as well, because it was mutating so fast. So this is how they would have detected different genotypes of the COVID virus. So in this branched amplification, and as it says in this figure, it is well suited for detection of nucleic acid target with sequence heterogeneity.
Hmm. So this is very specific for heterogeneity. They have
Yes, so even if there is, because the main structure of HIV virus, yes, you can detect that a person has HIV virus,
hmm.
but exactly what mutation has happened is what is important over here for
Hmm.
the disease. So it is going to check which genotype is present. So
Hmm.
this is really important for the genotyping. So I thought this figure was really neat. So
Mm-hmm.
what they are doing is there is a capture probe, which is right over here, which is black, which is again immobilized on probably a well of a plate,
Mm-hmm.
96 well plate or 384 plate.
Mm-hmm.
And then there is this capture extender, which is probably complementary to your capture probe. And now you have the target RNA or DNA, which is binding to your capture probe. Once you have this and you wash. It is like a, and all these signal amplification, I felt like this is the same process like sandwich ELISA, which is there where it is a
Hmm.
sandwich technique is being
Hmm.
used. So now this light blue strand, which is there, is your RNA, target RNA or DNA. And now you are putting a pre-amplifier. So this is what is specific to the genotype.
Hmm.
And why is this important? Because you can have multiple probes in a single test and know what is going on. So, for example, in one well, you can have one probe, and the other, you can have another probe. Or you can have multiple probes in one. Depending on which one it connects, probably they have a different fluorescence in each one, and you will know which one is getting amplified. So now this this dark blue part of the branch, the trunk of the tree, is
Hmm.
connecting to the target RNA or DNA. And then you have this green amplifier, which is the branches,
Hmm.
and this is connecting to your preamplifier. And then the label probe will be connected. So the reason this is important or advantageous is because it is very specific.
Hmm.
Because you have, again, because of the sandwich method, there it is very less likely that there would be carryover contamination, or the label probe will attach without it being very specific. So that is the point of branch DNA. And as you see, then there's a fluorescence. So we can read this together.
Hmm.
Capture probe is immobilized to the support, which is over here.
Hmm.
Capture extender is bound to the capture probe. The extender has sequences that are complementary to the capture extender. The target RNA binds if the sequence is complementary. The amplifier binds to specific sequence or RNA, which is over here. This then connects to the amplifier to form branches. The labeled probe then binds here and amplifies the signal. This will be useful in determining various genotypes for HIV and Hep C. Advantages, as I mentioned, there is less or almost no carryover contamination as it is very specific, and multiple capture and extender probes can be incorporated. So, for example, for genotype 1 to 10, you can figure out which genotype is there in one reaction from one specimen.
For that reason,
Uh-huh.
we have to design our like for every genotype, we have to design it for that
Primers,
particular.
yes. Yes, the amplifier would be
Hmm.
pre-amplifier, or the amplifier would be very specific to your genotype, which probably is probably a nucleotide or two nucleotide sequence is different.
Got that. Okay.
And this is from the book.
Hmm.
Again, try to use all the resources together.
Hmm.
And what it is saying is that additional extender probes bind to the target nucleic acid, which we discussed before, and then the multiple reporter molecules loading on the target nucleic acid with signals. And again, they're talking about alkaline phosphatase to give the chemiluminescent signal, and they're saying the detection limit is up to 50 targets mole per milliliter.
So
And they're
normally,
here.
most of the
Mhm.
cases we have seen like they are using alkaline phosphatase to
Mhm. Mhm.
capture this kind of signal.
Yes.
Mhm.
Yes.
Okay.
And this is again the same similar figure, but a solid support. Here
Mhm.
you have your capture probe and the extender probe,
Mhm.
and then you have the amplifier and the label probe giving you a signal.
Mhm.
So now let's go to again. So this one is for HIV and hepatitis C.
Mhm.
So now let's go to the hybrid capture. So hybrid capture is used for human papillomavirus, CMV, and hepatitis B virus. And in this, all these, I was checking because I forgot what these viruses are. These are DNA viruses.
Mhm.
So the DNA of this virus is going to connect to the RNA probe. So if you see in this figure, there is antibodies which are present, which are specific to this RNA.
Mhm.
These will connect to the antibodies. This when you have a RNA DNA hybrid, those would be very specific than a DNA DNA hybrid or a RNA RNA molecule. So when you have a DNA and RNA hybrid, it will connect to the antibodies. And once, as you see in the left second part of the figure, this is connected. And now this, again, like a sandwich ELISA, now because this is connected, this is another antibody is coming, and this will cause fluorescence.
Hmm. With that antibody, there has some.
This is the alkaline conjugate, right? Alkaline
Oh, okay.
phosphatase.
Mm-hmm.
Yeah, and that is a reporter. So that is the substrate is using up the alkaline phosphatase.
Got
So
that.
if you want, we can read this. Hybrid capture
Yeah.
starts with hybridization of RNA probe to the
Mm-hmm.
denatured DNA target. So the DNA is coming from the virus, from the
Mm-hmm.
viral specimen, which is
Yeah.
HPV, CMV, or hepatitis B. And the
Hmm.
RNA DNA hybrid is then bound to the hybrid specific immobilized antibody. So this I am imagining is in a 96-well plate, which is
Mm-hmm.
already immobilized with the antibodies.
Mm-hmm.
And now your RNA DNA hybrid is connected to the immobilized antibody. You wash the well multiple times. And now a secondary antibody is added, just like ELISA.
Mm-hmm.
And this will bind to the bound to alkaline phosphatase. So that is what that black dot is, Abu. And
Mm-hmm.
it generates a signal in presence of chemiluminescent substrate.
So, okay. So secondary antibody bound to alkaline phosphatase generates signal in the presence of chemiluminescent substrate. Okay, got that. That's the second part that it tried to attach. If I have a specific DNA of that virus, RNA probe will come and join with that. It will hybridize. Then the antibody conjugate will come and they will gonna attach, and then I will get my signal.
Yes.
Got that.
And then what they are talking about is cleavage-based assay.
Mm-hmm. Yeah, that's the number third for.
Yes. So what I have read in various books and in cap inspections which I have done,
Mm-hmm.
they talk about Invader assays. And Invader assay is a proprietary assay for factor V Leiden disease,
Mm.
and that is what this is. So I think before reading this, I never understood what the Invader assay is, but now I do.
Mm.
So it is specifically for factor V Leiden mutation detection.
Mutations. Got that.
So, and they also say it is for hepatitis C and hepatitis papillomavirus genotyping. So, in this case, you have your DNA with the mutation, you have another probe, um, mutation present cleavage. So, and there is a test probe with the mutation. So, if it is, let's read it over here.
Yeah, yeah, sure.
Invader assay exploit the substrate requirement of the enzyme. A cleavable substrate is formed by hybridization of two probes and a template, which is the top left. This structure will not form if the probe does not match the template. So, for example, if factor V Leiden mutation is present,
Mm-hmm.
all these three probes, the DNA template, the one probe, and this second probe will
Mm-hmm.
connect. And because now it has become a triple helix instead of
Mm-hmm.
the double helix, the
Mm-hmm.
cleavage enzyme is going to recognize this. And now it will digest it. So now mutation is present because of which you have gotten a cleavage. So now this part, which is the top part of the test probe with the A mutation, and there is a fluorescent and the quencher, right? The
Mm-hmm.
same thing like we have talked about in the reporter and the quencher.
Mm-hmm.
This is because it is able to connect. I think
Like they
there
will be
would
able
be
to
fluorescence.
recognize that
Yes,
sequence. They will be able to bind with them.
yes.
Got that.
So in the right part, if you see, there's a test probe which is A and C, and there is no cleavage because it did not connect.
Mm-hmm.
But in case of over here, because it is the exact mutation, there's a T over here, and in your probe there is a A, it was able to form a triple helix, which is recognized by the enzyme. And what this says is if the proper substrate is formed, which is on the left side of the diagram, the enzyme removes part of one probe and that fragment forms another cleavable complex with fluorescent labeled reporter probe. Repeated binding and cleavage amplify the signal. In plate format, only those wells with a template complementary to the test probe will produce a fluorescent signal.
Repeated binding and cleavage amplify the signal. In a plate format, only those wells with a template complementary to the test probe will produce fluorescent signal. Meaning, like, which one has the mutations? If it can bind, it will cleavage, and after cleaving that part will be bind with the fluorescence
The
and quencher.
fluorescent probe. Mhm.
hmm Then they will
And
be able
then
to.
you will get the signal. So just like the other one which we did the hybrid capture, only when there is a exact same mutation
hmm
that it will form, and you will get a positive signal.
Hmm, got that.
And this is the part of the about the probe which they are saying the second step. So you are putting that other probe, which is the FRET probe, is added that has sequence complementary to the cleaved signal probe. So
hmm
now this fluorescent and the quencher, the reporter and the quencher, this has complementary sequence to your cleaved probe, right?
hmm
And once you have that, it is going to give the reaction. The 5'
hmm
end of the FRET probe has a reporter molecule that is located in close proximity to the quencher. As a result, intact FRET probe does not produce signal. The signal probe, now an invader probe, binds to the FRET probe, producing an overlapping region. Again, it is by cleavage. And now that the enzyme is going to cleave this, it is going to cleave right here between fluorescent and quencher. And because this is quenched, this is cleaved, your fluorescent is at the bottom. You guys get that?
hmm yeah, yeah, got that.
So just to reiterate the process, there is a mutation. In case there is a mutation present, you it is getting bound, and
hmm
then you are putting this test probe which will
hmm
bind only if there is a mutation present, because it
hmm
is exact complementary to the
Yes.
mutation. Now the enzyme cleavage, it is very specific for recognizing the triple helix which is now formed.
hmm
Now it will cleave this, and now this part of the probe is in the solution. And now you put the second probe, which has a FRET probe, which has a fluorescent uh, probe, which has a fluorescence and the quencher.
hmm
Because they are close together, you will not get any fluorescence.
hmm
But because this probe, the cleave part, it will form like a triple helix again. And
hmm
now again, that cleavage enzyme, which we talked about, this is going to disconnect between cleave between the fluorescent and the quencher, and now the fluorescent molecule is available to produce the signal, and this will keep repeating.
hmm
If you see the right part of the screen, this
hmm
is the test probe
hmm
where you are not get you will not, it will not form a triple helix because the mutation is not present in the sample.
hmm Okay, got that.
And
Like,
this third one's,
last
go ahead,
step.
Abu.
Yeah, for the last step about the cleavage is the important part. If
Yes.
also important because if the fluorescence and quencher are together, they will not gonna make any
Hmm.
signal. But when
Yes.
the fluorescence will be cleaved, then
And
it
that
will.
will be cleaved only because this is the fluorescent and the quencher has a complementary sequence to the one which was cleaved. And
Hmm.
as soon as it combines, it becomes a triple helix.
hmm
The cleavage enzyme will digest it.
Digest the fluorescence and then we'll get the fluorescence. When it will be releasing, it will give us
Mhm.
the signal. Got that.
Yes. And now this I thought was so cool, the
Yeah.
cycling probe. And in this, what they are doing is that there is a DNA, RNA, and a DNA probe.
Okay.
So there is again a reporter on the 5' end and a quencher on the 3' end. Because of the mutation, or because again this is used for doing for um, antimicrobial resistance. So, for example, there's methicillin resistance gene in the S. aureus, like the MECA gene, or the vancomycin resistant gene, which is VAN A and VAN B in enterococcus, or herpes and histoplasma capsulatum. So what is happening is um, this is binding to the target, which is the DNA. So this purple is your DNA target, and now your probe is binding. And because
So we are discussing a lot of about probe. If you
yes,
wanted to simplify, like what we know about, like what do you think about probe? What is probe? So is
so a
it kind of
probe
like,
is,
yeah?
it is like a primer.
Mm-hmm, mm-hmm.
I
Like
think probe is just like a primer, but it has just
fluorescence kind of thing.
sequences. Yes, it has. For example, those molecular beacons are also probes.
Mm-hmm.
Um, so I think probe is like a primer, but it has fluorescent molecule attached to it.
Got that. So normally, as we discuss, like normally, I think from our previous studies also, like probe is kind of is it will be single stranded sequence of the DNA or RNA. It can be either of that. As we see here, it
Yes.
is a DNA, RNA, DNA probe. So it have to be single stranded, and it will be complementary to my sequence, my interested sequence, where I
Mm-hmm.
want this probe to bind. So,
Mm-hmm.
like I have to design this probe.
Yes.
Got
So
that.
it is basically, I think, an oligonucleotide, which could be single-stranded DNA or RNA,
Mm-hmm.
which has a reporter gene attached to it.
Got that. Like it will tell me, okay, my interested genes are over there or not.
Yes.
Got
So in
that.
this, it says cycling probe produces fluorescence only when RNA probe binds to the DNA template.
Okay.
So your, uh, for example, if you're considering the MEK gene or the VEN-A or B gene,
Mm-hmm.
that is part of the RNA part.
Hmm.
And S. aureus is a double-stranded DNA virus, or I know it is
Asorius,
gram positive. Yes.
Asorius double stranded or single stranded, right?
Yeah, and is it DNA or RNA? Oh, yeah, I do not know if it is RNA or DNA bacteria.
It's a DNA.
Okay, so
Double strand.
okay, so what they are saying is that, for example, you have the VEN-A or the MEK gene. That is what I understood. That is complementary to this part,
Mm-hmm.
and now you add RNase H, which will digest only when there would be a signal, right? So
Mm-hmm.
you have the mutation, or you have this MEK gene of an A, when B.
Mm-hmm.
So you are having this stretched out, the DNA, RNA, DNA probe. Now you add the RNase H, it will cleave the RNA part of the probe, because of which the reporter will be at a different area, and it would produce a signal because now it is not close to the quencher.
Mm-hmm.
So what I understood is that if there is no complementary sequence, again, you would have to be the one who designs the DNA, RNA, DNA probe. If the complementary sequence is not present, you would not get the annealing, right,
Mm-hmm.
of the probe to the target DNA.
Mm-hmm.
And you would not, if it is not annealed, it's not
Mm-hmm.
hybridized, it will not be cleaved.
Mm-hmm. Okay, got that. Hmm.
And then I just made this table.
Hmm. It was really nice. This table has all the um, added informations that is very important, will be very important uh, for both
So
of the exam preparation. Yes.
right. So in this, as you see, there is transcription mediated amplification, which we talked about, the NASBA and the TMA.
Hmm.
And as Abu said, in TMA there are two things: reverse transcriptase and um,
Um,
RNA
RNA
polymerase.
polymerase.
And
Hmm.
in NASBA, there's also RNase H.
Yes.
And this is for tuberculosis, chlamydia, HIV, and cytomegalovirus.
Hmm.
Then there was probe amplification. Does anybody want to guess without cheating? What were the probe amplification ones?
Yeah, so probe will be LCR, uh,
Mm-hmm.
Strand displacement, and the
Yes.
replicase, qubit replicase.
Very good, Abu.
I think for MB preparations, I think Sana will take the MB preparation. So they ask these questions like which one is probe amplification. So I think for that I needed to study that. So, yeah.
Good. So for probe amplification in the ligase chain reaction, you are having two sets of probes,
Mhm,
and it is checking the mutation. For example, single cell sickle cell anemia, the
mhm,
single mutation.
mhm.
What is, sorry, the spelling of created is wrong,
Yeah,
but
it's okay. I think they
you
will
are
understand. Yeah.
but you are so you have the probes.
Mm-hmm,
Probe will hybridize only if the mutation is exactly there,
mm-hmm,
and now the ligase will connect the probes, and because this probe is connected to a solid support, you will get a signal.
mm,
The next one is a strand displacement. This is for tuberculosis, chlamydia, and gonorrhea. And in this, there is a bump primer, if you guys remember, and there was
mm-hmm,
another primer which will get displaced. And because there is a nick, there is an endonucleoside,
endonuclear
a nick
site.
will be created, yeah,
Mm-hmm,
and the strand will be displaced. So
mm-hmm,
all this is the probe which you have generated, it is getting amplified. And somehow I think the probe is connected to the solid support, which is giving the signal.
mm-hmm,
The signal amplification, these were three. Let's try to remember by recall. It was hybrid capture,
mm-hmm.
uh,
Branch DNA,
cycling probe, branch DNA, and there was one more, cleavage based,
cleavage. Yeah, cycling
the
probe. Yeah,
invader.
cleavage based.
Yeah,
Yes.
yes. So in the branch DNA, this was for the HIV, HCV genotyping, and in this, there were in on the solid support, you had different capture and probe, and
Mm-hmm.
then you get the target, which then binds to the um, amplifier.
Yeah.
And this amplifier is the one which will have a specific gene for your genotype, and
Mm-hmm.
this will create signal.
Yeah. And they are very important for the like if it has heterogen genetic.
Genetic,
Yeah.
yes. Mhm. And in hybrid capture,
Uh,
you are detecting the RNA and the DNA.
hmm,
So you have antibodies which are connected to the solid support. The RNA is already there.
hmm,
And then you put the DNA of the virus, because it is a RNA DNA hybrid that is specifically binds to the antibody. Double-stranded DNA or single-stranded DNA or single-stranded RNA would not bind. This will be this will create a signal. You would get another antibody with alkaline phosphatase, and
hmm,
you will get a reaction.
hmm.
The
That's
third
good
one
now.
was cleavage-based Invader assay, which is for factor V Leiden mutations. And
Hmm,
in this, what is happening is you are getting a triple helix, which is specifically digested by the cleavage enzyme. And this will only bind when there is a mutation present.
hmm,
Once it is digested, the probe will connect to another probe, which you will put the FRET probe. And because this will form a helix again,
hmm,
now this triple helix is again recognized by the cleavage enzyme, and it will cleave the reporter and the quencher so that
hmm,
the fluorescence is present in the reaction. Cycling probe, in this there's a DNA, RNA, DNA probe,
hmm,
and in this, if it is exact sequence, and this is for methicillin resistance, vancomycin A and B resistant, antimicrobial resistance. Um, RNA is H is going to digest your RNA part of it, and again the reporter is released from the quencher, and your signal is produced.
hmm.
And that's the end of the story, guys.
Yeah, about this chapter, I think it was today's discussion was very in depth, and the thing that you we were discussing. So it has two set the questions that you asked, but for the second set, there has like the inner portions. So I think we were talking about the um, ligase chain reactions, Strand displacement amplifications, how many primer
Mm-hmm.
they normally use. They use two set of
Mm.
primer, but the second set of
Okay.
primer has the outer portions of the primer and inner primer, like outer and inner primer, that normally binds to displace the inner primer products, and then the DNA polymerase extends the complementary primer. I think Strand displacement here. So if we see here,
Which color?
uh,
Where?
the
Okay.
purple color. So we were thinking this purple color um,
Uh-huh.
is another different set,
Uh-huh.
um, but it's the like outer portions of the second set, something like that. They were
Okay.
telling this in the Lela book also, like
Mm.
second para of the Lela book. So
Mm.
they were telling that. I think today's discussion was very very interesting. I think it will be helpful mm, for everyone, both MB and SMB. Um, okay, so if any of the questions are coming from this chapter, I think we'll be fine. Um, yeah. Uh, uh, next, what is your thought? Which chapter should be covered? So if I try to uh, check the book, so um,
Can you share your screen so we
yeah,
can look at the chapters?
yeah, sure, sure. Just give me 1 second. I'm gonna um, find out the book and try to um, open it. So let's share. Okay. I think Kelly wanted to join, but I think this week she can be able to join. Hopefully, maybe next week she'll be able to join. Um,
Okay.
and um, if if you are okay, then I will try to share this because today's discussion was very interesting. So I think I'll share with them, those who are interested, so that they can see, and if they have more questions, we can discuss more. So I think first chapter, we already know, it's not much
Mm.
information. So there has some tables about these restriction enzymes, and that it will be good. Gene expression, this is okay. Uh, this is not important that much. Um this is also not important. Chapter 6 we just covered. This is very important.
Mm-hmm. Mm-hmm.
Um, Chapter chromosomal structure and chromosomal mutations, that it's not that much of important. I think only few topics. I think gene mutations can be one important, and the DNA sequencing. I think we already discussed about DNA,
Discussed it, right?
yeah,
Yeah. Yeah.
DNA sequencing in depth. So that's one
Mm-hmm.
thing. Then DNA polymorphism and human identifications. I think this is very important. I think we had
Mm.
confusions about one topics that
Mm.
regarding the STR. So this
Mm.
is one very important chapter, and 11, chapter 11, that is the detection of and identification of
Mm.
microorganisms. So this
Mm.
chapter is mostly, I think, we have to see all of the tables again and again to remember that informations. Um,
Mm.
I think uh, last week or before that we tried to discuss little bit, but I think we can discuss again. But then DNA based tissue typing, it is very important. HLA, there is one questions, I think Sana was playing for MB. So one topics is about the null um, um, what was the concept. So it has some of the topics that are very important. Um, like for the HLA testing, um, and this is the last chapter, that is for the management, like QC and QA. So I think these three chapters, this DNA based tissue typing, microorganisms, detection and identification of microorganisms. But you said you were reading the principle and applications of molecular diagnostic. That is the Nader Rifai book. That's
Yeah.
very in-depth book for molecular microbiology. And I think so this chapter I think will be maybe our next very important chapter, DNA polymorphism and human
Okay.
identification.
Okay.
Here there will have some of the concept about I think the um, like the um, STR typing and STR analysis. So I think it will be interesting. So next week we will discuss chapter 10, that is the DNA polymorphism and human identifications. Thank you so much everyone for joining, and hope to see you next week.
All right. Thank you, Abu.
Thank you so much. Bye
Take care.
everyone.
Bye.
Good night.